Good afternoon and thank you. It's not easy to be second after Debbie, especially since we are working with a similar but not identical adjuvant technology. We have the title here. The adjuvant is a strategic component in modern vaccine development, and what we mean by that is that we think it's very important that the adjuvant is present early on when you start vaccine development, because the adjuvant is the agent that together with the antigen will make the outcome of the vaccine in the end. I will start with a brief company presentation so you know what Iscom is in relation to Tcell and other companies, and then I will concentrate to talk about our variant of this technology, the MatrixM adjuvant. And I will try not to double too much of what Debbie has already said. But of course, there will be some similarities noted. The Iscom as a company is about 10 years old. We have a history of animal vaccines adjuvant focus. We have been working with animal vaccines for most of those 10 years. We have an ongoing adjuvant production and sales. We sell approximately 3 to 4 million horse doses of adjuvant per year. And our present capacity in production is in the order of magnitude of about 50 million human doses, if it's translated. And the method of production is scalable, readily scalable. Since 2 years ago, we shift focus. And we are now primarily focused on human vaccines. We have built our experience within the animal field. We will not leave the animal field, but we have entered into the human vaccines field. And we did this with our proprietary adjuvant, the MatrixM technology, as a base. We are located in Uppsala, in one of Sweden's largest biotech clusters. The company is quite small, only 25 employees. In November last year, we did an IPO, and now we are listed on the stock market, First North in Stockholm. We have a development pipeline and marketed veterinary products with Merck Intervet, Pfizer, and we have acquired, during these 2 years, some human vaccine partners. Johnson & Johnson, Tcell, Genosia, and the Jenner Institute are our prime partners. This is our human vaccines pipeline, and you see it probably better than I do. There are some influenza projects, malaria, HIV, and also some projects here with Genosia. And we have our own proprietary seasonal flu, which we started a clinical trial this spring. It's not finished yet. We are also working on another non-disclosed project. Some of these licenses are exclusive, but many are not. I will just make this clear that the MatrixM adjuvant is not equivalent or totally synonymous with Iscoms. Many of you may know about the classical Iscoms that were published in the beginning or middle of the 1980s, nor is it the same thing as the Iscom matrix. I will tell you quite shortly what it is. When you consider an adjuvant for future vaccines, they need to do something that the current adjuvants don't do. Current vaccines and adjuvants, they have provided mainly antibody responses. They have done a lot of good things, all the old vaccines, and they will continue to do so. But they do not really protect against the diseases that rely on cellular responses. Future vaccines will be pure products, much more pure than the past vaccines. They will probably be purified recombinants or quite low or moderate immunogenicity. And it needs an adjuvant like MatrixM that enhances immunity to a diverse type of antigens. Also, the future vaccines need to induce high levels of biologically active antibodies, and that's also a feature of the MatrixM adjuvant. But the most important thing for the future is to provide activation of cellular immune responses in order to protect against severe pathogens like malaria, RSV, HSV, CMV, HIV. There are numerous of agents. And the good thing about the MatrixM, as with the Iscom matrix, is the induction of a potent balanced cellular immune response, including cytotoxic T lymphocytes. Also, future vaccines must be produced timely and cost-effectively, and there the substantial antigen dose-bearing capacity comes in handy. The MatrixM technology is saponin-based. You have seen these particles already. Saponins are good immune modulators. I definitely agree on that, and that's the basis for the potent immune modulation we see in the Iscom matrix and with MatrixM. We also formulate these saponins into nanoparticles with cholesterol and phospholipid. And by doing that, you increase adjuvant activity somewhat, but above all, you increase the chemical and physical stability of this adjuvant. So this adjuvant does not really require an intact cold chain. We usually say that the stability in the fridge is about several years, at room temperature several months, and at 37 degrees several weeks. So it's a very stable formulation. Efficacy is established in animals and now lately also in humans. And we have GMP material available also. This is a summary slide of the technologies, because I know that it's easy to mix it up. What is an Iscom? What is a matrix? What is an Iscom matrix? And all the saponins. And from the beginning, the first invention was the Iscom. It means immune stimulating complex. And it's the same particle, the nanoparticle, but in this particle you also have antigens incorporated into the structure. And this was in the end of the 1980s, where we were talking about antigen targeting, multiple antigen presentation, and things like that. And this particle fitted with all the theories really on what is a good adjuvant or antigen formulation. So it took us quite a long time to realize that the matrix of this Iscom, just the saponins and the lipids, is an excellent adjuvant to mix with antigens in solution. It can be used as a standalone product. And this is more or less the system as Debbie talked about, her Iscom matrix and the antigen. The MatrixM is a somewhat more complicated product because it relies on two different types of matrix structures made with different saponin molecules. But there are some great advantages with this kind of formulation. I will come into that. I will give you some examples on how the MatrixM adjuvant work. But the formulation of different saponins in different particles, not mix them and formulate them in the same particles, that has increased our safety margin with about a factor of 10 in sensitive animal species like mice, for example, or cats. In non-sensitive animal species, of course, you don't see any major difference. You don't need to use more of MatrixM than the previous products. It's still as potent for immune stimulation. There has been a lot of work around the saponins, and we are talking about fractions and purified molecules. We are talking about fraction A and fraction C. Fraction C is the fraction of the saponins that is more QS21-ish in their structures and abilities. Whereas the fraction A is another saponin. It looks a little bit different, and it has other characteristics. The fraction C is the most adjuvant active of these two by far measures. It's a potent adjuvant for antibody responses. You get IgG1 and IgG2A. The fraction A in Iscom or Matrix is not a very good adjuvant for antibody production unless you have a very strong antigen. You will get IgG1 and a weak IgG2A response. So in that sense, it's an inferior adjuvant. But when you look at the T-cell immunity, it's quite a good adjuvant. Surprisingly high levels of IL-2 and interferon gamma are stimulated. And especially when you put these two together, they enhance the activity of each other. When it comes to toxicity, there is some toxicity in the C fraction. The fraction A is non-toxic, I would say. It's not toxic. And it's not really hemolytic either. Not that it doesn't matter at all, because when you formulate the saponins in particles with the cholesterol and phospholipids, the hemolytical activity is abolished. So that was the rationale for using these two fractions for us. And when we looked at the different characteristics of the matrix produced with them, we saw that immune modulation was different, kinetics of responses were different, possibly they target different systems, and also the safety was different. And you should bear in mind here that 50 micrograms in a mouse is a very high dose. It's approximately a human dose, so you shouldn't be scared by the figures. But if you mix matrix particles as separate entities, or you have exactly the same composition in the same particles, you see a quite different safety in the mouse system. So apparently, when you combine these saponins, you get problems. But as different particles, not. And also, the requirement for the more adjuvant active fraction C is quite low when you mix it with the matrix A. If this is the response with only A, matrix A, you see when you mix in 5%, 10%, or 20%, you reach the top level of antibody response that you would have gotten from the fraction C or matrix C alone. So you can reduce the amount of the somewhat more reactive saponin by a factor of 10 approximately. Now we will go through some studies. It will be antigens, will be influenza and rabies, and the animal species will be mouse, dog, ferret, and man. Starting with some flu studies in mice, we were using a Vaxigrip vaccine off the pharmacy, and we gave that alone, or with MatrixM, or with ASO3, or with aluminum hydroxide. The mice were vaccinated at day 0 and 21. Antibody response were assayed at day 28 and 21. And cellular responses were measured by antigen restimulation in vitro of spleen cells taken at day 42 or 43. And we were looking at cytokines in supernatant. So a very straightforward, simple experiment. Looking at the antibody responses, you see that, well, primary immunization enhanced IgG1 levels with all three of them, but with IgG2A in mouse only with the MatrixM and the ASO3. The same situation after booster with IgG1, and very similar also for the IgG2A antibodies. We even actually see a decrease in IgG2A compared to the Vaxigrip alone with the aluminum hydroxide. Looking at the T cells in the spleen, we see that the matrix formulations are quite active. We see high levels of IL-2, interferon gamma. But as we always say, this is a balanced T-cell response. We see also elevation of IL-4, IL-5, and IL-10, and particularly in the groups that have got MatrixM. With rabies antigen, we did, I will present 2 studies, one in mice and one in dogs. The one in mice is a dilution of a dog rabies vaccine into which we have mixed in MatrixM. So we have given 0.1 unit to a mouse. The requirement for a dog is at least 1 unit, looking at antibody responses. And the next study was done in dogs with a human rabies vaccine, because the animal vaccines contain aluminum hydroxide, and we didn't want to have another adjuvant here. We mixed a dilution also of this vaccine because it's quite rich in antigen, the human vaccine, compared to the animal vaccines. And we tested no adjuvant, 25, 50, or 75 micrograms of MatrixM. We were looking at antibodies in ELISA, but above all, we were looking at safety parameters in the dogs. And for that sake, we used 2 different kinds of dogs. Small dogs, 10-kilogram poodles, and the larger dogs were retrievers. First, looking at the responses in mice, we can see that after the first dose, the vaccine alone is the pale green, and the vaccine plus adjuvant is the dark green. We can see some increase in IgG1 antibodies, but primarily you see an increase in the IgG2A antibody. And the same situation after booster, you get enhanced response, particularly within the IgG2A groups. When we measure more specifically virus neutralizing antibodies, or at least antibodies competing with monoclonals with virus neutralizing activity, those response parallels very much the IgG2A increase you see in these groups. These are the small and the large dogs. The safety was assayed on day 0, 2, and 4. It was blood chemistry, extensive blood chemistry, hematology, antinuclear antibodies, and physical examination. And the antibody response, we took samples at days 0, 21, 28, and 45. I show you only the 45 data here. First, on the safety, all these doses were very well tolerated in both the small dogs and the larger dogs. In fact, the veterinarians work with this. They said that these vaccines were much better tolerated than the normal dog rabies vaccine, most likely due to the fact that the human vaccine is a more purified antigen product. You can also see a very clear dose response with the addition of the matrix. I should point out that the purple lines, these are really, really high titers. So, in ferrets, a study with influenza. This is they were given 3.75 microgram dose of hemagglutinin, neuraminidase, or a combination of the 2 molecules, with or without MatrixM adjuvant. They were vaccinated twice, days 0 and 20, and they were challenged at day 52. If you look at the response, we can see that the virus replication in the lungs was substantially reduced, where we had a good response to the hemagglutinin in the hemagglutinin plus matrix or the hemagglutinin neuraminidase and matrix groups. Substantial reduction of virus replication. Looking at morbidity, which is also important, we see that the response to the neuraminidase is the most important one. We have the neuraminidase plus matrix, or the combination of hemagglutinin and neuraminidase plus matrix being the groups with the lowest morbidity. And if you look at lung lesions, the quality of the lungs after infection, you will see that, well, both the responses to hemagglutinin and neuraminidase make a difference. We have hemagglutinin plus matrix, neuraminidase plus matrix, or both molecules plus matrix. So now to human studies. We have only been in the human area for 2 years, so we haven't done very much yet compared to what CSL has done. But there are some studies that have been completed and some data is coming out. The first data is generated by the PanFluVac group. It's a EU finance consortium within the 6th framework. They did a study with the MatrixM and avian influenza vaccine. It was a virosomal formulation of the hemagglutinin mixed with MatrixM. It was a study containing 60 subjects, adults 20 to 49 years old, 2 intramuscular doses, days 0 and 21. The first group got only 30 micrograms of hemagglutinin, no MatrixM, and these 3 groups got 50 micrograms of MatrixM mixed with 1.5 microgram of hemagglutinin, or 7.5 or 30 micrograms of hemagglutinin. Endpoints were safety, local and systemic reactions, and efficacy, of course. If you just summarize the results first, there were no severe adverse events, and the adverse events that were noted were mild and transient. And all CHMP criteria for hemagglutination inhibition were met in all matrix groups after the second administration of the vaccine. T-cell responses were augmented by the MatrixM, and the formulation was definitely dose-sparing. In this table here, you can see the 30 microgram doses without or with MatrixM was a primary response, a secondary response, and the lowest amount, 1.5 microgram of hemagglutinin with matrix after the second dose. And you can see that this is a really competitive formulation. The low antigen amount plus matrix is almost in the same level as the 30 microgram dose and significantly better performing than the 30 micrograms without adjuvant. Looking at the T cells, this is influenza-specific CD4 T cells. You get from the 30 microgram dose without adjuvant some cells, but already at the 1.5 microgram of hemagglutinin with matrix, you get a larger number and increasing up to the highest dose here. We wanted to do something on our own as well, so we have initiated a clinical trial on seasonal influenza in elderly. The study was started in May, June 2011, and it's still ongoing. We have sampling days 0, 28, 90, and 150, and 150 will be by the end of November this year. We took a marketed inactivated split virus influenza vaccine and simply adjuvanted that with our MatrixM, a 50 microgram dose. And we did a pre-study in adults looking at evaluating the safety before we entered into the main study with the elderly subjects. So these were vaccinated and the safety was evaluated, and after that we could go into the main study, consisting of 2 groups, one group that received the flu vaccine only, and another one, the flu vaccine plus matrix. Endpoints are safety, of course, and efficacy, hemagglutination inhibition, virus neutralization, homologous and heterologous, and cellular responses like cytokines and granzyme B will be measured. Whether these are the right things to measure remains to see. But the study is still blinded and ongoing. It's very exciting. I will finish off here with some mechanism of action. We have very similar findings than that you have at CSL, of course. The MatrixM is not a depot adjuvant at all. As Debbie said, most of the action is in the draining lymph nodes. And the physical interaction between the antigen and the adjuvant is not mandatory, but they need to be given together or at least so they are drained into the same lymph node. We don't see any pronounced pro-inflammatory activities, and that's also very similar to the data that Debbie showed on the cell lines. Also with blood cells, we cannot really see strong pro-inflammatory activities going on by just mixing the adjuvant with cells. And we also see very similar adjuvant activities in all or many species. You can recognize the typical signum of this adjuvant in cat, dog, horse, fish. If you give the adjuvant alone, no antigen mixed in, you will see that a lot of cells are recruited to the draining lymph nodes. These are fold change of different cell types. So you see it's all cell types, T cells, B cells, phagocytes, monocytes, dendritic cells, NK cells, and of course a lot of neutrophils. And when we first saw this, we were quite excited and thought, well, this is why this adjuvant works. You can also see that the amount of cells going into the draining lymph nodes, it happens quite early in mice with 3 micrograms. If you give 30, there's a very small increase in the numbers. But we have seen from a lot of studies now lately that this seems to be a very typical finding with adjuvants. You see it with MF59, ASO3, even alum. And these are very different adjuvants. So there must be something more happening that is crucial for the adjuvant effect. And we did some studies in the pig. These are very recent data. They were first presented last week on a poster in Seattle. We have truckloads of data, but I've just put up a smaller summary here in the end. We gave MatrixM intramuscular to pigs, and we've been looking at the genes affected at the site of the injection and also in the draining lymph nodes. And to summarize only on the surface of this study, we can say that MatrixM preferentially upregulate interferon-regulating genes. You can see a substantial amount of the genes upregulated are somehow involved in interferon regulation. Also, MatrixM modulates the interferon response and expression of pattern recognition receptor genes. As Debbie said, there is no interaction. We have not found any interaction with Tcell receptors either. But apparently, we see an upregulation of TLR2 and 4 at the site of injection. Also, the MatrixM affects the expression of cytokines and chemokines. There's a whole list of various cytokines and chemokines affected by the MatrixM. But I will stop here, just conclude that the MatrixM adjuvant is a potent and useful adjuvant for increasing qualitative efficacy of a wide range of vaccine antigens. Main features are the antibodies and the cellular responses, the typical balanced T-cell response, CTLs. It's very simple to use. You just mix it with your antigen. Our variant of this technology, the MatrixM, has just entered into human clinical trials. You have the PanFluVac study where data is coming out, and we have our own ongoing study for the moment. And hopefully also in the not too far future, we will see results coming out from studies performed by our partners. Thank you so much.
Thank you. Are there any questions for?
went long ways to sort of split up saponin into individual components and then pick A and C or C for the Iscom matrix. Is it so difficult to synthesize this, or is this still a very complex mixture, this fraction A and fraction C? Because the obvious thing to do would be, okay, model that nature gives the model, and we now synthesize it and make an entirely synthetic particle.
There is a group that actually has synthesized at least the QS21 major molecule. I think it took 5 years or something like that. And you know, none of us has shown the structure. The structure of these saponins looks like cholesterol with branched carbohydrate side chains. So it's a very complicated task to synthesize this in the right way. Maybe in the future you can do some sort of simple version when we know how and why it works. Maybe we can take the groups that are essential only and make something. But I think it will be quite expensive also to do it on a totally synthetic base.
Just to be clear, so what you have as a fraction A is still a mixture. It's not a defined molecule.
No, the fraction A is not a defined molecule, but it's not a bunch of molecules either. It's a family of similarly related molecules with very similar effects, different effects to other saponin families found within the extract. But it's not a single molecule. Neither is QS21, even if it's a much narrow peak, because these molecules they have a life. You have side chains that are migrating within the molecule, so they are fairly complicated. But with purification, you can cut down on the varieties and focus on those with your specific activities that you require.
Thank you very much.